I just wanted 1 or 2 slides there. Okay, so I'm here. I'm going to minimize that. I'm going to come back. Yeah. So as I was saying, no, so we have restated the problem now, and what we are asking is the question.
So I guess your screen isn't being shared.
Oh, okay, good thing. I just was in a rush to do it. Let me know if you see the screen now.
Yes, it's visible.
Okay. So what I was saying is that, given that the problem we have here, where we are just basing this analysis on the flux analysis, and what we're saying is that if we restate this problem, then what we're asking is, what is so special about this pathway that makes it difficult for us to produce methionine? Given that the wild type is producing a very small amount, we have developed a strain that produces quite a reasonable amount, but it still is far from that 19.23 gram per millimoles per gram dry weight per hour of flux that we looked at. Whereas this is close to this concentration as well as this concentration as we see. There's a variation of about 3 times. So unless we increase this by 3 times, we can't really make it economically feasible. Okay. So obviously the answer is deeper than this. And what we notice at the outset when you look at this network is that for sleek glutamicum, for which this strain was produced, where obviously if you had auxotrophs, then this lysine and threonine is absent. You have to supply it from the outside if you want, because cell growth is dependent upon these 2 components also. Whereas by itself, it will not produce lysine and threonine. So that's the meaning of auxotrophy. Auxotrophy means that you've essentially mutated this by some method, lysine and threonine. It could be random mutations, and then you select out the strain that is not producing lysine and not producing threonine, or you essentially use a cloning method by which you cut off this pathway and this one so that these 2 components are not produced. And so you have a straight pathway going down to methionine. So it's very important to notice this because there is a regulation that is very strict here. So the regulation is that from lysine, which is the very first important amino acid that is being produced, you have feedback regulation right onto the first step right there. Threonine has a feedback to that lysine, that first step of this semialdehyde. So there, and then also at this step here. Sorry, this is aspartate synthase. So these two steps are going to be modulated by this. We also produce isoleucine. Isoleucine is also going back and inhibiting the first step. And L-methionine, which is being produced here, is repressing this step here. Okay. So there's a very strict regulation, as you can see, and that's because there are 7 ATPs required for a synthesis. And once you commit for this and you spend the 7 ATPs, the cell does not want to retract that step. So it wants to make sure that the decision it's taking is correct. So these are all these regulatory steps are there just to ensure that the decision making process is regulated properly, so that you don't commit to producing methionine. Now, obviously, methionine is very important for 2 reasons. One, it is required as the first amino acid that goes into the protein synthesis step. And secondly, it has many other functions. And I've not shown all the other steps that are there on the side here for within the cell. So it is an essential amino acid. Human beings don't produce this. And we have to obtain this from other resources, either animal resources or plant resources. So this amino acid is not synthesized by human beings in the natural metabolism, but it has to be obtained from an external food source. So keep that in mind. So what we do is we essentially break this wiring diagram into steps that will help us to look at what we call the enzyme block, the gene block, and aspartic acid. So how do we go about really creating this whole thing. So obviously I have this model, but this model is of no use unless I know the reactions that are going into this block here, right? So unless I know what are the reactions inside this and how I'm coding for this, there is no way I can really make this useful. So I have to know the parameters. I have to know the rate equations. And then I have to build the gene block. That means, what is happening at the level of the gene. So that's the level of the enzyme. So but this enzyme is being synthesized by a sequence, as we are considering very simple ones here. We're looking at the operator block, the RNA block, and then the enzyme synthesis block. So three small steps that are considered here. So we kind of simplify it and just break it down to these three steps. So, and obviously in that case, then the enzyme that we normally assume to be constant in a biochemical pathway is going to vary depending upon what is the regulation that is taking place. Clear? So those are the two things that you have to keep in mind. Now, how do you start doing all of this work? So I'm going to now bring in the Excel sheet that I used to kind of create this. So close this out. So you need to look at, so what you need to do whenever you do a project like this, you will essentially have to list the name of the enzyme. So aspartate amino acid, so the kinase, so the amino acid that is, so aspartate is the first compound. It is an amino acid, and the name of the enzyme that is going to act on this substrate is aspartate kinase, and the EC number for this is 2.7.2.4, and this we can get from the Keck pathway. So if you go to the Keck pathway, you will find that this value comes for this particular thing. So the molecular weight is given. You look at the reaction. So now I've broken up in two parts. The blue part is for the gene level, and this orange part is for the enzyme level. So you have two different components that you're dividing. So now you look at the reaction. So the reason is, see, when you're gathering the information, it's important that you collect it all in one place, so that later on you don't have to go back and forth. And if you've forgotten something, you don't have to go back to that page, because on the internet surfing and getting all the information down is a lot of time required. If you put a student to do this, it'll be a complete summer project just to get this information out. Okay. And then you make your own personal comments, whatever you wish to write. In this case, I've written the enzyme is for E. coli and is a multifunctional protein which catalyzes this reaction. And I've written it for E. coli, and maybe I don't remember now. You pick up one value because maybe the C-glutamicum value is not present. That's the reason the E. coli enzyme was picking up. So what is the signal it takes? What is the regulon it is on? What is the gene that it's acting on or is concerned with? Then what are the transcription factors that are which are present in this case? Does it have a function? What kind of things that are there? So all of those things will be additional information that you keep. So the 4P signaling that is there, and all the information that is required for other additional information that is required is all kept here at the gene level. And then you look at the substrate. So S1 is L-aspartate, substrate 2 is ATP that is required. Then you look at the product. The first one is L-phosphate, aspartate L-phosphate it will be, so 4-phosphoaspartate, so L-aspartate. And then the product here, second product is ADP, because that is the pair normally that we see. And then you look at specific activity. You look at other things that are important here. You look at the VM value, KM value, and you look at if there's any inhibition that is there. If there is an inhibition, then you have to know what is inhibiting it. So inhibited by inhibits aspartate substrate inhibited. I wrote here, so you can read the full thing here. So repressed and not inhibited aspartate substrate lysine is inhibitory for streptococcus mutants, and that value something is given as 10 there. And then this is also some additional information for the wild type for S. cerevisiae. So, but it's not inhibitory for so it will have inhibition taking place from aspartate, but and other information related to inhibition by lysine and Streptococcus and other mutants given. So we could not get the data for this. Maybe we use some of this data. So I have to go back and see. But so there's been so those things you have to remember. I can't remember that. So now let us look at. So I go through all of this, and then let me go and show you the MATLAB. So what I'm telling you is that when you're doing the Excel sheet, just so be careful. You put in all the data once and for all. So and depending on the project, and depending on the information that you require, you can make these columns as you wish, and then fill in all this data, and you can put in the source where you're getting it from. Also, you see, there's another thing that I need to show you, is that there is a reference that is given here. So keep these references also, because finally, if you're submitting a paper, then that research paper that you're going to write will require you to have this reference. So whatever be the source that you got this information from, just mark it down somewhere so that later on you don't start hunting for this again all over again. So do the work once and for all. So think through the problem entirely. Imagine that you're going to do some great work, you're going to publish, and then you're going to put this in a good journal. So all that information is collected in one shot. Okay, just give me a second. All right, sorry about that. I'm in the office. This is what happens. So can't switch off that. Okay. So any questions up to this point?
No, sir, it's clear.
Clear?
Yes.
Okay. So this is just demonstrating how you would be gathering all the information and collecting it and keeping it together in one place. Now, how do you use it? So one of these references that you see here, some of the references will actually give you some idea if somebody has in the past done some modeling or not. Else you'll have to search and make sure that you are either using it and acknowledging that I'm using this model, or you say that well, based on these models, I'm developing my own equation, or based on the KEGG information, I'm developing my own equations, right? So whichever. So you do that. Then let's come to this here. So I'm going to take the simplest of these cases here so that I can illustrate what's happening here. So look at this. So this is the gene level. So I'm just having an input in this. I'm just put one here. Then I look at the function that is there. This is an embedded MATLAB function, and within that you can have other units that are present. And so in this case, I have only one equation. So one enzyme, and therefore, so there is an S dot, which is the rate of change of the substrate that's coming in. If there's any change in that, if not, that rate equation is given, and then the product of the equation is given here, and you have all the inputs that are shown here. Okay, which is the parameters that you require for doing the integration and computation for this set. Okay, so that's. I don't want to stop this. I just want to minimize that. Okay, maybe I'll just go back from here. It's easier. Okay, let's look at something a little more complex. So you see inside this is the gene level work that I showed you there. So for the operator component, you'll find that you'll have one set of equations. So the O is for the operator. Then for the RNA, you'll have another set of equations. So mRNA, rate of change of that is given here. And then the enzyme. So E dot, the rate of change of the enzyme is given here. Okay.
So where do we get these values of these parameters?
So that's what I showed you in the Excel sheet. I showed you. So you go to KEGG, and from KEGG, you'll have other resources. Then if you can't get the information that you need, you'll have to hunt. And I've given my remark here. So inferred computationally without human oversight. I don't know why I wrote that anyway. VM and KM values are given for this. And maybe there's something else missing there anyway. So no entries for E. coli and VM. So I had to pick it up from some other source. So I've marked out so let's say this one. So I have another comment here. So inhibitors, D-lysine and are not relevant to in vivo. So these are all comments that I have. So, and this is the two enzymatically active forms of L-lysine tRNA synthesis. So there are some papers which are there. So it's a lot of digging around. That takes a lot of time sometimes. Until you're satisfied that you're getting the right, or at least the ballpark values which are all consistent. So you might, if you get an option between E. coli, suppose you're doing for C glutamicum, and you have an option between E. coli and yeast, it is better for you to take the E. coli because yeast will have a variation. It's a level above the bacteria, right? Or if you're doing if you have mammalian cell data versus let's say yeast data, then you might take the yeast data because mammalian cell data will be far from C glutamicum, right? So some decisions like that have to be taken, but idea is that you'll have to fill up all these columns. So these kinetic parameters have to be filled up. So see there is written here, see no data for E. coli, so I picked it up from some other. So all right.
Yes, sir.
So you have to pick up all of this from the KEGG. Mainly, the KEGG will give you most of it. If not, there are now so many other sources from where you have to pick up this data. Okay, and of course the equations you can write your own equations, or you can see if there's a model existing and then use the help of that model and acknowledge that model if you find that is a good model. If not, you can always go by the first principles and you write your own equations. That's not a problem at all, actually. Okay.
Yes, sir.
So and all these connectivities are there based on the wiring diagram. Okay. So depending on the inputs and outputs, you can just keep on building it, and based on those inputs and outputs, you'll get cues on the side here. For example, if I delete this, right? Oh, I can't do it now because I have actually kind of run this whole thing so that I can show you some data. Okay, so let's look at aspartate phosphate. So I think it has run it. I hope it has run this. Let me see if it gives me the no, it's not yet run. So I'll have to run it one more time, maybe continue. I think I must have stopped it somewhere in between. Okay. So that is the way the aspartate looks. Okay. There's a spike and then it goes down. If I were to look at, let's say, homoserine, so that's the way it varies with respect to time. So this is all in seconds. And this is, I forget the units, must be millimoles, I'm not sure. So let's look at
the initial variable that is being modulated.
No, so what you do is you consider, so what you say is that, well, I'm going to look at how this network is going to behave based on some variation in the aspartate, because that's what you want to check, no? What happens when there's a change in the aspartate that is coming into the flux. So the flux in the aspartate will change when there's a cue from the cell, some signaling from the cell that tells it to change the aspartate concentration, right? So you can take, and these values that I put here are the one or whatever the units will be here, will be based on the cellular concentration. So I just change the cellular concentration by maybe just a small amount and perturb this network. Then the perturbation can be of various kinds. So in this case, I have not shown all of the. So there's a constant, there's a ramp, there's a step down. You can also have a step up. You can have pulse. I've done all of those. I have not shown them here actually. So you can have pulse. You can have a sequence of pulses. So or step up, step down. So many things you can do, and all of those are comes from the library. If you go to the library and you look at sources, then you'll find, see, this is the source for various kinds of inputs which are here. So you can have pulse, you can have ramp, you can have repeated sequences, you can define so many of them, right? So you have a step up, you can have a sine wave. So depending on the problem, you define one of these, or you define your own repeating sequence, by yourself. You can define it. Or you can pick up a signal from from a source. You can copy the data from another resource and just put it as a signal here. All of that is possible. Do you see that?
Yes, sir. So, but like, how do we get to know that what is actually happening inside the cell? So, like, is the aspartate changing in like a linear fashion, as in a ramp change, or is it changing in a step fashion? So, how do we get to know that?
No. Yeah, no. So that would be it would be based on an experiment. So this is based on the experiment that I had actually put a pulse of cysteine, right?
Right.
So, or if I put another pulse, so I put a pulse of glucose, or I put a pulse of aspartate in the experiment, right?
Okay. So that pulse or that ramp input is given like externally, not from.
That is, if you had done some experimental work, you can do that. That is external, or you assume that is what you're doing for the, or you can say that this is a hypothesis. I'm going to test out this hypothesis. I'm doing this simulation, and I want to see what I get, and then I'm going to do an experiment and see if this matches this or not. Or you can go the reverse direction. You have a set of experiments, and you want to verify whether the output is giving the same result or not.
Right, sir.
Clear? Vishantan?
Yes, sir. Yes, sir.
Okay. So and this one is, then you can see, let's say for example, the homocysteine. So that's the variation in the homocysteine, and then the methionine. The methionine becomes constant here. So the important thing to note, actually, that actually what we found out from this thing is that once all this is committed, after a certain point of time, methionine should be produced steadily, right? That's our inference, because now it is going to be required for cell synthesis, protein synthesis in the cell and other cellular functions. So the initial input will only affect this magnitude of response. This also, there is a saturated maximum level up to which it can saturate, beyond which it will not produce any further, because that we know that as we saw, 1 to 2 gram per liter of methionine was something that you could get maximum given this network. This is not the complete network. So you can see that becomes steady, right? And this is a good thing, because that's what you are expecting. That once the commitment has been made by the network, it should be producing at a regular output that can be used for other processes. And so that is what the demand shows. You see? Also, I did a few other things they're not shown here. You can also look at the tRNA of synthesis of all of these important components, and you'll find there are perturbations in the tRNAs of other components, but not in methionine. Because once you've committed, there will be slight variations that are occurring there because of this perturbation, but nothing will happen to the methionine. And this was tested out. We've done some experiments and tested this out, so it works out. Okay.
Yes, sir.
So this is how you would do this whole thing, and you can see that the entire thing is quite an elaborate thing that happens there. But keep in mind that this is only for these 10-11 steps that we talked about, right? And that becomes so complex. So therefore, the Simlink has a limitation. Keeping track of all these, it's a good thing to start off with. If you know broadly what the gene regulations are, and you can do that. But if you were doing a large project where you, let's say, have 100 reactions or something like that, perhaps it's better to do some coding. Okay. So, and that's what we found. So we have other papers which we have done where we looked at bigger networks, and in those cases you have to do coding rather than do it on Simulink. But Simulink is a very good, useful tool for illustration, for teaching, as well as for understanding. Whenever we're doing something small, it's a very nice tool for understanding, because you just map the wiring diagram directly into this kind of small toolboxes and just add the edges very quickly. As long as you have the data, very quickly you can see what's really happening. So that's the usefulness of having the Simulink. Okay. All right. So I think this should be fine. If all of you agree, then I'll start the new lecture. Is okay?
Yes, sir.
Yes, sir.
All right. Not much time is left, though. I can see that we're already in 46. But anyway, let me do the due diligence. So I'll just give an introduction of signal transduction. I will not start the lecture, but I'll just talk about it, and then we'll talk about the importance of this, and then kind of give you start the lecture in the next class. So signal transduction, if you remember, when we talked about evolution of the cell and life forms and other things. The most important step where we know that, it perhaps was the first step before actual life took place is that enclosure of all these, compounds, or rather all these components, not compounds, all these components like the rudimentary RNA, rudimentary proteins and metabolites to be enclosed by the cell wall, right? So there was some kind of a cell wall, some kind of a lipid bilayer that was there, and that essentially enclosed the cell. Now, by doing this, it did two things. It essentially gave the evolution a chance to create the first living organism, a cell. But at the same time, it also made the cell isolated from the environment, right? Now, for continuity of the cell has to respond to the environment. And therefore, the cues that come from the environment are going to be molecules that affect the cell's, nutrient, if it requires nutrient to propagate, other things, so whatever, for the cellular processes to continue and to continue as an organism and create a new organism, right? So there are, of course, many missing steps which we are not aware of, but idea is that there is this lipid bilayer which essentially blocks off the environment from the intracellular milieu, right? And the process by which the cell essentially communicates or receives the communication from the environment to within itself is called signal transduction. In the context of the cell, signal transduction in the broader context means changing the signal of one kind to another kind. So, for example, I could look at resistance, the change in resistance, and I could map that to temperature, right? So signal of one kind, which is change of resistance, is getting transduced to the signal of another kind, which is temperature. So that would be a signal transduction that takes place. So at the level of the cell, what it means is that you're getting a molecule to sit on the receptor on the outside. So there's a ligand that comes and sits on the receptor, which acts in the similar way and gives a new signal within the cell for it to react, right? So, for example, if you have adrenaline coming on, binding to a receptor on the cell surface, immediately the cell will produce some secondary messengers within the cell to react to that, so that maybe a lot of oxygen can be taken in, or depending on what kind of response it requires, or a heat shock response. So some protein which is a heat shock protein binds to the cell, or some cue which talks about the environment being changing because of the change in temperature. The cell will then immediately start pumping out maybe fluid from the cell, or do some other things that will essentially make sure that the cell has a chance to survive in that environment. So that is what essentially is the whole process of cell signal transduction. So this is the first layer of information that the cell receives. So the reason why we're jumping back and forth here is because we can't do the transcription part of it unless we understand what signal transduction is. So we went bottoms up. That means we talked about enzymes and how enzyme kinetics are important to a cell. And then we talked about the response that we see from the hill kind of response that is there, which is like a switch, and that has many other important features that we'll talk about later on. And then we moved up one level and we said that, well, that the synthesis of the enzymes are being controlled by the regulon or the operon, which is there at the level of of one single enzyme, and the regulon would be many such enzymes being controlled by one logical unit. So that is the way we are looking at the cellular system right now. So we had come to this level, and above this would have been well, if you're looking at the synthesis of or rather the translation of a gene into a protein. Well, we could have discussed that right now, but really it goes hand in hand with why would it start to respond. So there has to be a cue for it to come, and that cue comes from the signal transduction, signal that comes from outside that goes inwards, changes something, and then gives you a need for this transcription. So here we'll have to go from the top down again so that we can kind of make it logical. So that's the reason why we are doing the signal transition first and not transcription networks first. Okay, so then the whole thing will tie up together. So I'll stop here and I'll take questions if you have any questions right now. And we need to talk also, when is the minor beginning?
The 14th to 17th.
And when is our official minor date?
17th.
So I guess it's 6. Yeah, 17th.
17th. Okay. So anyway, so most probably I'll give you the paper on that date, and then we will take it on the and receive it back on the 22nd, which will be the class. So 22nd, 22nd, I might be late for the class because there is a faculty interview which has been fixed without my. I don't know. I'll have to see the date time. Maybe it's in the afternoon. If it's in the afternoon, is fine. Oh, we are having on. Okay, Tuesday we are having on at 5 o'clock. So it should not matter. All right. So if there are no further questions, then I'll just conclude for today and see you guys on Friday.
Sure, sir. Thank you, sir.
Okay.
Thank you.
All right. Yeah. Bye.
Thank you, sir.
Bye.
Thank you, sir.